Hi, everyone. I welcome everyone who has joined in for this webinar. At the outset, I'd like to thank ISCH and Professor Mohammed Mohti for giving us this opportunity to present our work. I am Dr. Hasmukh Jain. I'm a professor in medical oncology at Tata Memorial Hospital, Mumbai, and I'm here to share our learnings from the research that we did in India on CAR T cell therapy. So I'll essentially take you through the background, set the context of what has happened and why this is important. This CAR T-cell therapy research that happened, which led to the commercialization and approval of the first CAR T-cell therapy in India. I'll take through the journey of what happened from the bench to the bedside and eventually to the commercial access. I'll also talk a little bit on how we as clinicians and also our partners are looking at improving the access, and then a little bit about the next steps and future directions. So these are my conflicts of interest. Now, the most important thing to understand first is where India stands in healthcare. Now, India, as you must know, is a big country. I mean, it has the largest population. We have 1.4 billion people. It's also one of the countries with the most rapid GDP growth at present. So we have an annual GDP growth of about 6 to 7%. But unfortunately, of the entire budget that we set, we only assign less than 2% of our budget for healthcare. So healthcare is a big challenge for us. And even though India is known as the pharmacy to the world, being one of the countries which does the maximum production, maximum manufacturing of drugs, we still lag in innovation. We did demonstrate our potential during the COVID crisis, where we were able to provide vaccines not only for people in the country, but also to several other countries who did not have this manufacturing capabilities. But innovation is where we lag. And something similar to what happened in the space race and the space story, where India was late to join the space club, but it is well and truly arrived. This probably could be the moment in cell and gene therapy where India has just started to catch up now. Now, one of the biggest problems that we have in India is a limited access. Now, we know that there are six approved products from US FDA, but the prices of these therapies are remarkably beyond the reach for most of our patients. And just to bring this to context, the average per capita income of an Indian is about $2,000, as opposed to $30,000 or above for most of the people in the developed countries. And to say that a therapy would cost close to $500,000 places it beyond the reach of 99% of the patients in this country. So that's a big problem. And the only way of solving this problem would be if you innovate. And why is the need to innovate? Is because of this. India has the second largest number of cancer-related mortality. Close to 1 million deaths because of cancer happen every year in India. And this is primarily because of the delay in access to most of the innovative therapies. Typically, there is a lag. The lag could range from anywhere from 5 years to more than a decade for any latest drug to be available to India. And this is true also with CAR T-cell therapy. And this is not only true for India, this is also true for most of the LMICs. As you can see in the cartoon to the right, that most of the LMICs don't have an access to CAR T-cell therapy. Of course, these are also the regions which actually are in need for such innovative therapies. And we know that CAR T-cell therapy is an innovative therapy. It has been given the breakthrough innovation. It has changed the treatment paradigm in leukemias and lymphomas, but yet it remains beyond the reach. And the products that are already available will not be accessible to most of our patients because of the exorbitant drug cost and lack of insurance coverage. As you can see in the graph here, less than half of the patients in low, mid, and low income countries actually have insurance coverage, and that is true even for India. So to summarize the challenges that we are faced with is that in a setting where we have high incidence of cancer, a high number of patient population, high cancer mortality, we need access to innovative therapies. What is already available is beyond the reach of our patients. So we need to innovate. And to innovate, we need complex technical skill set. You need complex clinical training. You need an infrastructure, very important, particularly for cell and gene therapy. You need dedicated GMP facilities. There are challenges in terms of design, in terms of construction. We faced these challenges when we were doing our facility construction, because this is completely novel, and so we had to, in fact, take assistance, also look into the smaller details of how the facility is constructed. And there are regulatory challenges for being the first cell and gene therapy which is approved in India. Even the regulators were not ready, and so it took a lot of time and a lot of a lengthy process for us to get the approvals going. But this was possible because of a collaboration of some of the biggest institutes in this region, and this happened basically by as a result of a chance meeting. So, as clinicians at Tata Memorial Hospital, me and my colleagues were looking for scientists who could work with us on this particular project. So we were searching actually all across the country and also for global partners, only to find that the innovation is actually happening in our backyard. So Indian Institute of Technology is one of the premier engineering institute in Bombay, which is just about 20 miles away from Tata Memorial Hospital where I work. And and just on a Google search, we were trying to find out who was working on CAR T cell therapy way back in 2013, and we found this presentation from the lab of Dr. Rahul Purwar, who was working at IIT Bombay, and he was also working on CAR T cell therapy, and then we started reaching out to him, and this led on to this whole journey. And we were supported during this journey by various collaborators, national as well as international, particularly National Cancer Institute, who was very helpful during the whole journey. And this has led to this construction of this beautiful facility called ImmunoAct, which is now able to cater, has the capabilities to cater to about 300 to 500 patients per year, and they are rapidly increasing the scale further. So this is a summary of the journey. This is about a decade of research, and we started in 2013. The initial few years were spent in developing the lentiviral platform and the CAR T. Then the next set of challenge was to develop the vectors in sufficient batches so that we could start the trial. We started the phase 1 clinical trial in 2020 after receiving this approval from our regulators. And then subsequently in 2022, late 2022, we started the phase 2 trial. In August 23, we enrolled the last patient. And after 3 months of follow-up, median follow-up, we got the market authorization, becoming the first product which has received the approval first cell and gene therapy product that received the approval in India. So this is the product that was tested. So this is a humanized CD19 CAR T cell therapy product, and I will tell you a little bit about why humanized and why did we choose humanized. At the time when humanized was still not the popular choice, all the commercially available products at that time were murine, the time when we started our research. So as you can see in the cartoon here, the linker is a CD19 directed antibody single chain variable fragment, which has a hinge domain. There's a transmembrane domain, and there's a co-stimulated domain. So we worked on both the CD28 as well as the 4-1BB, but the one we chose for the further clinical trial was the 4-1BB co-stimulated domain, given the fact that there was data to say that 4-1BB perhaps will lead to a lot better persistence. This product has been patented, so the Indian patent is granted. It is also the US patent is still pending, and this is the one which we tested in the further trials. And as I was saying, that when we did a preclinical work, what we found, if you can see the cartoon, the figure B in this particular slide, when we looked at both the humanized as well as the murine product, what we noticed is that while both the humanized and the murine products were working similarly in terms of cell kill, but when we looked at cytokine activation, so when they were co-cultured, when the CAR T cells were co-cultured with this tumor cell line, we found, and in the presence of monocytes, we found that the activation or the release of interferon gamma and interleukin-2 from the monocytes was much lower when we used the humanized product, as opposed to using a murine product. So this is one of the advantages of having a collaboration right from the beginning, right from the stage of preclinical work, because the clinicians were collaborating with the scientists, and because we were very clear in letting them know that we have a high patient burden. We would need something which is going to be less toxic, at the same time have equivalent efficacy with murine. And with this idea, the scientists took this forward, and they selected the murine product for our further development. And the similar activity was also demonstrated in the animal models. So here you can see in the animal model study, what we did is we took not-skid mice. They were irradiated. The tumor cells were then inoculated. We selected the CD19 expressing NALM6 tumor cell line. And then the CAR T cells were infused on day 1. And subsequently, bioluminescence imaging was done starting from day 11 at weekly intervals. What we demonstrated, as you can see in the figure at the bottom, in the control population, the tumors, there was a proliferation of the tumor, whereas in the CD19 CAR T treated, that is a humanized CAR T treated animals, we did not see proliferation of the tumor. The tumor burden was substantially low. So this again showed that this CAR T cells worked even in the animal models. This work was published in the Molecular Cancer Therapeutics. In fact, the cartoon that you see on the cover page of Molecular Cancer Therapeutics in that particular issue is the depiction of our CD19 humanized CAR T cell structure. And this work was also referenced in the prestigious Nature Biotechnology magazine in December 2019. So with this information, we knew that we had a humanized CAR T, CD19 directed CAR T cell therapy product, which demonstrated robust anti-tumor activity and at the same time had low cytokine production. We hypothesized that this could be something which could translate to lower side effects in the clinics, and this is what we took forward into the clinics. This slide, just to summarize the kind of regulatory approvals that we required, and this is typical of any cell and gene therapy product. There were approvals from the biosafety committees, from the animal ethics committees, then our institutional ethics committees, and the CDSU, which is our FDA equivalent. And so we started the process. The process what we followed is something which is very well established globally. So we will do the apheresis of the patient that's where the cycle begins. So we select the patients, enroll the patients under the clinical trial. We do the apheresis, and then the product gets manufactured transported to the manufacturing site. Once it reaches the manufacturing site, over a period of next 6 to 9 days, using a semi-automated process, they took the product went through the various steps, starting from T cell selection and activation, lentiviral transduction, and then followed by QC. And once the HCAR19 expanded, harvesting of the product, followed by cryopreservation. The product would get released when the patient was ready at the clinical trial site. So this is a standard process that we followed. Now, a brief summary, and I'll share the further details later. But a brief summary of this phase 1 study was that we were able to treat 10 patients. And what we demonstrated was we chose a wide range. I mean, we knew that it is very difficult to identify dose levels in CD19 CAR T cell therapy. This is because the absolute numbers in the product will be different from what eventually happens in vivo, depends upon the level of expansion. So we said that we will test the dose range and then try to identify what is the safe dose and also figure out what is the effective dose. So we said 10 to the power of 7 till 5 times 10 to the power of 9 CAR T cells would be the range that we would start on. We used a fractionated dosing strategy to ensure that there's safety. So the CAR T cell would be given as 10%, 30%, and 60% on 3 consecutive days. We selected the lymphoma patients in the initial trial, and the patients were selected as per the SCHOLAR-1 criteria, which meant anybody who had a best response as stable disease of progression to the last line of therapy were enrolled onto the trial. Now, the key finding was that we treated patients at doses that range from dose as low as 2 million cells per kg body weight to as high as 17 million cells per kg body weight eventually. 7 out of these 10 patients got a dose of more than 5 million cells per kg body weight eventually. And what we demonstrated was that across the spectrum of doses, there was no treatment-related mortality. There were no ICU admissions. There was no neurotoxicity, importantly. And there was absence of grade 3, grade 4 CRS. This is a surprising finding at that point of time because murine CAR T cell therapy, you expect a high incidence of ICANS, even if it's grade 1, grade 2, and you also expect some level of grade 3, grade 4 CRS. But subsequently, this was also validated by other researchers who are working on humanized CAR T cell therapy, and they demonstrated that perhaps the humanized CAR T cell therapies are much safer, at least in terms of lower neurotoxicity and lower incidence of cytokine release syndrome. So I'll take you to the study design of this of the phase 1 as well as the phase 2. The phase 1 was a single center open label trial, and we recruited 10 participants. We gave CAR T cells over a wide range as a split dosing fashion, and this was for patients with lymphoma. Moving on to the stage 2, this became a multi-center open label trial, and we recruited 50 patients. The dose we identified for the phase 2 was more than or equal to 5 million cells per kg body weight. This was based upon the previous experience. Now, here we also included patients with leukemia. That's because the target was the same. And we had some experience from the pediatric trial where this was shown to be equally safe. So we selected a dose of 5 million cells per kg body weight as the starting dose. And we nested a cohort of 3+3 for the leukemia population. So we said that we will first test the 5 million cells per kg body weight in the leukemia cohort, and only if it qualifies as safety, then we will continue enrolling at the same. Otherwise, we will go a dose level below. So patients in this study were 15 and above, and the study was conducted. These are the steps in the study. So we screened patients, we found eligibility. We found the patients who were eligible and then enrolled them onto the trial. The first step would be apheresis. Bridging therapy was allowed in this particular trial, and the bridging therapy for the lymphoma was left to the clinician's discretion, depending upon, of course, what therapy was received and whether the patient was symptomatic or not. In the leukemia, we defined a few regimens. Some of them were low-intensity regimen, like giving 6-MP methotrexate, versus some of them were high-intensity regimen, like the hypercivadin modification. The conditioning therapy was a standard fludarabine cyclophosphamide. So we, after the first few patients in the phase 1 trial, we settled on a regimen that was used throughout, where fludarabine was given 30 milligram per meter square, cyclophosphamide 500 milligram per meter square for 3 consecutive days. After the rest of 2 days, we infused the product at a dose of more than or equal to 5 million cells per kg body weight. Patients remained hospitalized for 7 days, and then there was a weekly follow-up for first 28 days, and then subsequently the interval kept on increasing. So it was 3 monthly for 2 years, and then 6 monthly, and a mandatory follow-up till 5 years. This is as per the regulatory requirement to look for the late complication. Now, the response assessment was done on day 28, which was in the form of a PET-CT for lymphoma and a bone marrow study for leukemia. So this is a summary of what happened with the patients. So 64 patients were enrolled all in all, 14 from the phase 1 and 50 from the phase 2. If you look at disease subtype, 17 patients were leukemia and 47 were lymphoma, including 4 low-grade lymphomas. Now, out of this, 62 patients were leukapheresis, 57 were infused. So more than 90% of the patients who were enrolled did receive infusion, which is comparable with what is already seen on other trials. And 10% attrition is expected even before they get infused. That's because of the nature of the patients we select on these kind of trials. These patients would have aggressive disease, and there is a possibility that they would not remain fit for the infusion. Eventually, 53 patients qualified for what is known as the efficacy evaluable cohort, which was defined as patients who received at least 5 million cells per kg body weight. Now, the 4 who did not receive, 3 of them were from the phase 1 trial. That was a part of the design, because we were at that point of time still finding the dose. And 1 patient in the phase 2 did not receive the desired dose. So here is a summary of the patients who were enrolled. So the most common patient cohort was the high-grade lymphoma cohort, and the next one was BALL. The age was there was a wide spectrum of age. So we had patients still about 71 years who were enrolled. Predominantly 3/4 of these were males, and patients had received at least 2 lines of therapy. Just to impress upon the kind of patients who were selected, these patients represent what is truly seen in our clinic. So, for example, if you look at the lymphoma cohort, 1/4 of these patients had bulky disease. And if you look at the number of extranodal sites, again, almost half of these patients had at least 1 extranodal site at the time of enrollment. Similarly, LDH was elevated in most of these patients. So the average LDH being 257, the upper limit of our range is 190. And similarly, if you look at the leukemia cohort, the average blast percentage was 61% at the time of just before the infusion, and it was as high as 98% as well. So really representative patients were enrolled on this particular trial. Keeping that in mind, if you look at the results, the overall response rate was the primary endpoint, and we demonstrated 70% overall response rate, which therefore we were able to reject the null hypothesis. Our hypothesis was that from a baseline of 25% in this kind of population, CAR T-cell therapy would improve the outcomes to 50%. So we were able to reject the null hypothesis. And when we looked at the cohorts individually, both lymphoma and leukemia, we found that the response rate was similar. So it was 68% in the lymphoma cohort, and it was 73% in the leukemia cohort. These are representative cases of to show the activity, and this is in lymphoma patients. As you can see, the first patient to the left had bulky disease in the retroperitoneum, and this patient had a CR on the day 30 scan. And the second patient is the patient who had a primary refractory disease, who had failed on R-CHOP, had a growing tumor bulk. And on day 30, he had a CR, and he's also the patient who is at the longest follow-up. So he's just recently crossed 2-year time point, and he's doing well. So to put things into perspective and to see what is how does our data stand, at least with this initial results, when compared to the other approved therapies. So if you look at the efficacy perspective, and particularly in the lymphoma cohort, you will see that you'll appreciate that the ORR is 68%, somewhere in between what is reported with T-cell, Iso-cell, and then Axi-cell. When you look at the leukemia cohort, again, it is somewhat similar to what is reported with the other products. Of course, the limitation of the sample size. remarkably, I think the standard feature is the safety profile. So when you look at the adverse events of special interest, what is important is that neurotoxicity was not seen in any of our patients. Even the cytokine release syndrome was predominantly grade 1, grade 2, and only 3 patients had grade 3 cytokine release syndrome, and that was only in the leukemia cohort. So there's no grade 3, grade 4 cytokine release syndrome in any of the lymphoma patients. And that is again that again stands out when you compare these results with the other approved products, whether it's the CRS incidence of CRS, I mean particularly the grade 3, grade 4 CRS in the lymphoma cohort or in the leukemia cohort, it is much reduced. Particularly the CRS grade 3, grade 4 CRS in the lymphoma cohort is definitely reduced when compared to the murine products. Now, neurotoxicity is a standard feature, whether it's leukemia cohort, whether it's lymphoma cohort, there was no neurotoxicity, and this is something which is remarkably different from the other approved products. So here I summarize the neurotoxicities, and we put this data in context with the other approved products. And as I said, the neurotoxicity and lower incidence of cytokine release syndrome is something which is the most standard feature here. So all of this translates ultimately into a lower utilization of healthcare resources, and one of the most important things is access to or the utilization of ICU. So as you can see here, the only 7% of the patients actually required an ICU admission, so which is again remarkably lower when compared to the other similar trials, and also in terms of other aspects of healthcare utilization, so resource utilization, particularly usage of tocilizumab, usage of steroids, this product was at least similar when compared to the other products. The need for vasopressor was much lower, obviously because of the lower incidence of grade 3, grade 4 CRS. And the difference was similar even with the leukemia cohort. So this work was presented at ASH. This was an oral presentation, ASH 23. My colleague, Dr. Athar, did summarize these findings nicely. So what we did highlight was that the need for ICU admission post-infusions is very low. And if you look at most of the pivotal trials that led to the approvals, at least 30% of the patients required ICU admission, which is much lower in our experience. So that is, this is very relevant. So I suppose this is relevant for even the other regions, but particularly more so for the LMICs, because the moment you have lower toxicity, that means that there's a lower healthcare resource utilization. It reduces the cost of clinical care, which is another important component. We always think about the cost of the product, but healthcare utilization cost is also equally important. And then because of all this, it improves the accessibility, particularly in our settings. We have exploratory translational work, which is a part of this trial, and this is still not complete data because we are still analyzing our data. But from the limited samples that we've looked at the CAR PCR expansion, we have demonstrated that the dynamics of CAR expansion mirrors what is already known. So we see generally the peak in around the day 14, and then there is a decline around the 4th week, and then beyond that it declines, and then it becomes gradually it is undetectable. At very low levels, it is found even at month 6 in many of these patients. So with this product, and this product was approved in October 23 by our regulators, and now this is the first CD19 CAR T-cell therapy product that is available. But of course, this is just the beginning for us, and we know that the world has moved far ahead from CD19 CAR T-cell therapy and looking at different options. What is in pipeline for us as Tata Memorial Hospital as an academic center, and also our partners ImmunoAct, which is now a commercial entity, is to look at how to improve the outcomes of CD19 CAR T-cell therapy. One thing which is now nearing approval is to test CD19 CAR T-cell therapy in combination. Because it's a humanized product, it is allowing us to combine with other drugs, and without enhancing the toxicity. We hope it doesn't enhance the toxicity, but we at least have that the courage to go and try to do combination therapy. So we're looking at combination with nivolumab and or pomalidomide. So it's a phase 1 trial at this point of time. We are also the work is also going on in terms of trying to test this molecule in CLL. So we could use a higher dose in CLL, because CLL generally the activity of CAR T-cell therapy has not been great, probably because of the poor quality of T-cell. So we feel that one is by increasing the dose of T-cell, the CAR T-cell therapy, we could overcome this to a certain extent. But more importantly, by then testing in combination with molecules like BTK inhibitors and PI3K inhibitors, we could probably improve the outcomes even in CLL. So this is the second research which is being planned. Besides, the preclinical work with BCMA CAR T cell therapy has completed. So we are in the process of getting a phase 1 trial activated. This is again going to be humanized BCMA CAR T cell therapy. There are other things in the pipeline like CD9 triple CAR T cell therapy, which is targeting CD19, 20, and 22, and some of the targets for solid tumors. Now, the next challenge for us as an institute and for our partners is how to improve the access. Now, the work started in this small lab, which is about 750 square feet, which is 150 square feet lab for the manufacturing and the QC lab, which is good for the phase 1 trial. But now, to scale up, we have this beautiful facility at ImmunoAg, which has the capability of handling about 300 to 500 patients. But even that will not be sufficient. It will not be sufficient even for one institute, Tata Memorial Hospital. And remember that this now we have to provide not only to one institute, we have to provide to the entire country and also possibly to this region, so the South Asian region. And so therefore, this is the next challenge that we're facing, and we are constantly trying to improve the access. What we have done so far is, when you look at Actalicel, which is the name of the product, Actalic Atvogen Autoleucel, is that we are able to reduce the cost. That's mainly because we control the IP. So the patent is with an Indian researcher. Because the process of manufacturing is an integrated process, and it is not a completely automated process. So it allows us to scale up. And because we propose to follow a regionally decentralized model, wherein what we propose to do is to have multiple centers in different regions, so in the north, south, west, and east, which could then cater to the patients in that particular region. So you have a central facility which does the vector manufacturing, which does the training, but your regionally decentralized, regional centers who then do the final CAR-T manufacturing, and therefore expedite and scale up the process. So this, we feel, is one of the most viable approach, particularly in a region like ours. And this is what is being planned to improve the access and scalability. So, as I said, we should be able to reduce the cost. Presently, the cost, the price marketed for this particular molecule is roughly about 40,000. It's going to be about 40 to 50,000 US dollars. But depending upon the requirement, we've been promised at least that the cost could reduce down to $25,000. And one of the reasons is because the IP is owned by in Indian site, is vector production capabilities, which is actually a bottleneck, is also controlled by them, and they're able to manufacture the vector on their own. And the manpower cost is something which is in control. So the manpower cost in our region is definitely much lower when compared to the other countries in the West. So eventually, not only the manufacturing cost, because of the reduction in the toxicity at different phases in the therapy, pre-infusion, peri-infusion, and also because of the lower utilization of the healthcare resources, eventually it is likely to reduce the cost and improve the access. So with this, I'd like to conclude, and these are all. I would like to acknowledge the role of all these people who are involved in this trial. So members of my group from across the specialties who help us throughout the process. The cartoon, the image here in the middle top. In the upper middle is the day when we did the first infusion. This is way back in June 21, and then this in the figure to the left bottom is the commercial team, all these young scientists, and the team who's involved in the manufacturing, the quality control, and they were all excited on the first day. On the day the first product was shipped out. The work has received a lot of attention. You see the figure in the middle, the bottom figure. None less than the president of the country invited my collaborator, and this work was appreciated. And also, this has received a lot of media coverage. So I would like to also acknowledge the participation, the contribution of several groups of people. It's not possible to list each and every one of them, but I've tried to list out the groups, particularly a special mention to the mentors who have helped us, like Dr. Satyanila, Dr. Nirali Shah, and Dr. Nitin Jain, who have helped us guide through the process and been patient enough to listen to us when we ask them, our regular questions, our emails, do multiple calls with us, and help us take through the process. And ultimately, finally, last but not the least, the patients and the families who placed their who participated and placed their trust in us during this research. So with this, I conclude. Thank you.
I think I've responded to two questions that we put up. One was about the impact of metabolic tumor volume. So yeah, we have seen that there is a negative correlation between the metabolic tumor volume and the outcomes in the context of lymphomas. Um, Then the second question was really in relation to whether if immunoglobulin replacement was given if the patient had less than 400 IgG level before the infusion. Yes, so if the antibody levels were below 400, then an IVIG was administered to patients even before the CAR T-cell therapy infusion was given. What's the explanation of the absence of ICANS? I think one of the strong reasons we feel is it's because of the design of the construct. It's a humanized CD19 CAR T-cell therapy. And with the humanized CD19 CAR T-cell therapy, this particular neurotoxicity has been reported much lower. And it's true in other groups as well who are working with this with the humanized CD19 CAR T-cell therapy. And I think that is the biggest reason why the neurotoxicity was less. Okay, so there is a question of how many centers in India have started CAR T-cell therapy apart from Tata Memorial. Roughly about 10 to 15 centers have been. So the way it has moved on was that on the clinical trial, there were a total of 5 centers who were working on CAR T-cell therapy with us. And once the commercial approval has come, so every site or new sites are getting added on, and there is a little bit of an accreditation process, a training process. So in that sense, about 50 to 20 centers have become a part of this. And some of the centers have also started doing few cases. Most of them are in the very early phase, but about 15 to 20 centers have been identified and who have been prepared to do the CAR T-cell therapy in the future. So what support is available to patients who can't afford these therapies at this time? So, I mean, there are different ways it is being approached. So in Tata Memorial, we being a central government institute, and the philosophy being that patients who cannot afford need to be supported. So we go through various governmental as well as non-governmental agencies to raise support for these patients. And we have we've just started looking at the various options or various NGOs who would support this kind of an effort. Because from our transplant experience, we know that it can take a while before you are able to raise the funds. And CAR T-cell therapy more so, the time becomes very critical because you don't have much time to organize. So we've just started in the last 3 months. We have started doing this off trial. It's been encouraging. I mean, we've been getting some support both from government as well as non-governmental agencies, but there will be still a lot of planning and a lot of support which will be required for us to be able to treat patients to kind of match up the capacity. So this is a work in progress. I mean, there is a question on how to put an order to procure CAR T cells from you for our patients. So the way this works is CAR T-cell therapy at this point of time can only be done at a center which has the training and the infrastructure available to do CAR T-cell therapy. The manufacturing team is a separate team. This is a company started off by our scientific partner. So they do this whole process of accreditation, legal processes, etc. And once that is done, then they have a certain manufacturing slots. At this point of time, they are scaling up, but at this point of time, they have a limited number of slots. It's about 15 per month. So you are expected. So once you become an accredited center, you share the reports of the patient. I mean, share some of the basic details of these patients, and then they prioritize and then they give the slots according to their availability, or let you know whether a slot. They tell you the average waiting time for these kind of situations. So that is how the process starts. And then there are, of course, various other regulatory aspects and legal aspects that will be discussed once the center comes on board. So what is the first step to build a CAR T-cell infusion center? Is it necessary for a center? Not necessarily. I mean, if you have a transplant unit already, that's already a plus. That already means that you have the necessary infrastructure, which is little more than what is actually needed to do these kind of therapies. But it's not necessary for you to have a HEPA filtered separate unit. What is essentially needed is a good transfusion medicine support, a good intensive care support. You should have the ability to simple things like be able to thaw the product bedside, have a biosafety cabinet to handle the product bedside. and also have the necessary experience and the protocols, because this is different. So I think the key difference between a BMT versus a CAR-T is that initial myelosuppression is actually not very profound. So it's not so much of handling this myelosuppression which is a problem, and therefore it's not necessary for you to have. It's good to have a HEPA filtered unit. It's not necessary though, and in a single clean room you can start off these therapies. But I think training, understanding the protocols is the most important thing, because that is a little different from what we do in the regular hematological malignancies as well as BMT. Are the products homegrown, or they are sent and brought from outside? I don't understand what that means, but I suppose you're trying to ask whether this is manufactured within the country or this is just a licensed product, and whether the product is imported. So no, this is completely a product which has been developed here from end to end, including from the viral vector till the manufacturing of the CAR-T and the quality control. So the product is manufactured in India. So specifically at this point of time in Mumbai, the only facilities available in Mumbai. So yes, so the product is shipped to that facility. They manufacture and then they ship it back with the quality control results. So besides Mumbai, people have been. I mean, the centers have shipped the products from Delhi. They've shipped from South. So those SOPs of logistics and shipping has been sorted, and now there's sufficient experience on handling this, and handling the logistics part, I meant. What is the turnaround time for the humanized product? So the turnaround time as of now is 3 weeks. Generally, it takes 2 weeks for the product till the time the product is harvested. It takes another week for the QC to be run, and the final for us to see whether the release criteria is met or not. And then, so essentially, a third week is when the product is released. Are there local trials applying CARs for solid tumors? So there was work happening with one of my colleagues who was working on GD2 CAR. That is still a work in progress. My scientific partner has been working on a few other products, particularly in close collaboration with UPenn, and then a couple of other centers as well, but nothing as yet in the clinics. Are you open to transfer the technology as an academic project to developmental country with no access to commercial CAR-T? So I essentially do not. I mean, I as in Tata Memorial Hospital doesn't own the patent on the product. This is, as I said, with the scientific partner, so Dr. Rahul Purwar, as I mentioned during the talk as well. So ImmunoAct is the agency that holds the patent. They are definitely open to this idea of collaborating with LMICs, and the whole idea, as I said during the talk, was to improve the access to the LMICs. There's already been a an MOU collaboration initiated with Mexico. So this is definitely something which is on, and this is something which they'll be very interested to do. So please feel free to reach out to Dr. Rahul Purwar. You can always also write to me, and I can connect you to him. My email address was mentioned on the presentation, so you could definitely do that. And yes, you could look at the possibilities there. Are there any other centers in India who manufacture CAR T-cell products? I mean, there are at least a couple of groups that I'm aware of who are working on CAR T-cell therapy. They're essentially, the model that they are working is licensing products from other regions in the world. So there's a group that is working with Spain. They have got a license to procure that product to India, and they're working on that. There's also another group which is getting the CD19 from, or the BCMA rather, I think, from China as well. But there is no, till date, there's no other group which has done the whole manufacturing in India. So yeah, so that's the difference. And I see that's the end of the questions. Okay, thank you. Thank you so much.